Welcome back to what I did in lab yesterday, where I tell you guys what I did in my biochem lab. This time I have an update on my previous lab as well as my current lab. First of all, this is how the negative control for the mint plants in the rooting media looks. You can see it's quite a bit less fluffy than last week. There's less of that really fluffy white fungus, and now it's condensed and compressed quite a bit more, and there's a very thick, sludgy black layer on it. I'm not entirely sure what that is. Again, it might be bacterial, it might be fungal. Either way, it's pretty gross. This is one of the positive samples. You can see it's growing quite a bit better. There's a good amount of leaves growing out of it, and if you remember from a good few weeks ago, these started out as naked stalks with just the node and no actual leaves on them. So now they're growing quite a bit taller and they have a bunch of leaves on them. It's hard to see again in this picture, but you can see right down there is a root growing. It is kind of hard to see, so I promise there's roots there, which is good because that's what we want. Here's another picture you can see a little bit better. On that short one here, you can see it's not really growing many leaves, but it is growing a slightly longer root, which you can kind of see. It's a bit out of focus, but it is growing a decently sized root, while the other one you can see is growing a bunch of leaves on it, which is good. That's what we want. Now for the regeneration media with the lavender plants, it's grown quite a bit. There's quite a lot of that really gross looking green mold on it, and the really shiny juicy orange stuff is kind of being taken over by all of it. There's a lot going on in here, but you can still see that one lavender leaf is hanging on. The rest of them, you can see they are way overtaken by mold and everything. That little guy is still going. Very impressive. Here's that same plate, but from the back side. I just thought it looked cool. It's way lighter from the back. It's not got that green layer on top as much, and you can kind of see the lavender leaves a little bit better. I just thought that was cool. Now, if you remember from last week, we did have that one weird plate that has those black dots on them. We decided ultimately that it wasn't contamination because it wasn't growing. There was just black dots on there. Nobody knows what it is. My TA asked me if I sprinkled black pepper on it because that's what it looks like. So that's still there this week. But also we did unfortunately have one small dot of contamination up here. That looks like the very starting of one of those orange batches of the slimy bacteria or something. So that's probably going to continue to grow, unfortunately, but that's what it looks like right now. But as for the rest of them, excluding that one at the top, which is the negative control, none of the other plates were contaminated, which is good because at this point, almost all of the other groups had at least one plate that did have a bunch of contamination showing up after the 2-week mark. Ours, other than the negative control and that weird speckly one, they're uncontaminated, all of them, which is great. We're one of the only groups that have no contaminated plates, so love that for us. Now, it's too early in the lab progression for this actually to happen, but the end goal of this is for these to form calyces, which is a bunch of plant material that will grow into the new plant. That's what the regeneration media does. It forms these calyces on the ends of these lavender leaves. And so if you wait long enough and then eventually plant them, they will turn into brand new lavender plants. The lab won't run that long with these, so that really won't happen with the lavender plants. But if you were doing this in an actual biochemistry lab setting, where your end goal was to make a bunch of these plants, then that is what would happen. As for the updates on the other lab we're doing, the Agrobacterium transformation, we have our carrot slices that have been cooking in the Agrobacterium for a week, so ideally, all of the Agrobacterium would have transported the GUS genes into the carrot cells by now. Now, I did say last week that these carrots would be blue, but they're not blue yet. They will be blue next week. So what we did this week is we took all of the carrot slices and we kind of wiped off all of the excess bacteria. Then we cut them into kind of wedge shapes, about 1/8 of the entire carrot disc, and put them into these tubes containing the Gus assay. And the Gus assay is what will trigger the blue color. In a couple weeks after the carrots have been soaking in the Gus assay, the negative control carrots will show nothing because they don't have the Gus A gene, but hopefully, if done properly, the Gus positive carrots will show this very nice bright blue color. Hopefully, if we did everything right. But as of right now, this is the before picture. This is after they've been cut up and now they're soaking. One of them is the Gus positive, one is the Gus negative. So these guys will stay in their tubes for another few days. Then when we come back next week, hopefully the carrots will be blue. But until then, this is the update we have for the labs.